Hello everyone, and welcome back to another chapter of Advanced Pathophysiology. Today we're discussing chapter 21. These are the mechanisms of hormonal regulation. So, this looks like a relatively brief chapter. So, without much preamble, let's jump right into it. The endocrine system is composed of various glands located throughout the body. These glands synthesize and release special chemical messengers called hormones. In addition, numerous hormones are synthesized and released from endocrine cells within the upper and lower gastrointestinal tract, your incretins. The endocrine system has five general functions. So here we're taking a quick peek at the major endocrine glands. So, our five major functions: the differentiation of the reproductive and central nervous system in the developing fetus, the stimulation and sequential growth and development during childhood and adolescence, the coordination of male and female reproductive systems, which makes sexual reproduction possible, maintenance of an optimal internal environment throughout the lifespan, and finally, the initiation of corrective and adaptive responses when emergency demands occur. The endocrine, nervous, and immune system work together to regulate responses to the internal and external environments. Hormones convey specific regulatory information among cells and organs and are integrated with the nervous system to maintain communication and control. The mechanism of communication and control occur within a cell, the autocrine, between local cells, the paracrine, and between cells located remotely from each other, the endocrine. So, mechanisms of hormonal regulation. Endocrine glands respond to specific signals by synthesizing and releasing hormones into the circulation, which then trigger intracellular responses. Although a wide variety of hormones function within the body, they share certain general characteristics. Your hormones have a specific rate and rhythm of secretion. Three basic secretion patterns are the circadian or diurnal patterns, your pulsatile and cyclic patterns, patterns that depend on the levels of circulating substances, your calcium, sodium, potassium, or the hormones themselves. Number two, hormones operate within a feedback system, either positive or negative, to maintain an optimal internal environment. Number three, hormones affect only cells with appropriate receptors and then act on those cells to initiate specific cell function or activities. And number four, your steroid hormones are either excreted directly by the kidneys or metabolized, conjugated, by the liver, which inactivates them and renders the hormone more water soluble for renal excretion. Your peptide hormones are catabolized by circulating enzymes and eliminated in the feces or urine. So hormones may be classified according to the structure, gland of origin, effect, or chemical composition. The secretion and mechanism of action of hormones represents an extremely complex system of the integrated response. So again, we have our peptides here, your growth hormone, insulin, leptin, parathyroid hormone, and prolactin. Your glycoproteins, your follicle-stimulating hormone, your luteinizing hormone, and thyroid-stimulating hormone, and peptides, your adrenocorticotropic hormone, your antidiuretic hormone, calcitonin, endorphins, glucagon, your hypothalamic hormones, your lipoproteins, your melanocyte-stimulating hormones, oxytocin, somatostatin, thymosin, and thyrotropin-releasing hormone, and amines, your epinephrine and norepinephrine. And then we jump into the lipid-soluble, your thyroxine, then your steroids, your estrogen, glucocorticoids, mineralocorticoids, progestins and testosterone, and finally your derivatives of arachidonic acid, your autocrine and paracrine action, your leukotrienes, prostacyclins, prostaglandins, and thromboxans. So what is the regulation of hormone release? Hormone release occurs either in response to an alteration in the cellular environment or in the process of maintaining a regulated level of certain hormones or certain substances. Hormone release is regulated by one or more of the following mechanisms: your chemical factor, such as your blood glucose or your calcium channels, the endocrine factor, a hormone from the endocrine gland controlling another endocrine gland, and neural control. An example of chemical regulation is seen when insulin is secreted following chemical stimulation by increased plasma glucose levels. Cortisol from the adrenal cortex is an endocrine factor that regulates and stimulates insulin secretion from beta cells within the pancreas. Neural control occurs when the autonomic nervous system directly stimulates the insulin-secreting cells of the pancreas. Your feedback systems provide precise monitoring and control of cellular environment. Both negative and positive feedback systems are important for maintaining hormone level within the physiologic ranges. Your negative feedback is the most common and occurs when changing chemical, neural, or endocrine response decreases the subsequent synthesis of secretion of a hormone. Positive feedback occurs when a neurochemical or endocrine response increases the synthesis and secretion of a hormone. The next figure illustrates both positive and negative feedback within the hypothalamus-pituitary target organ axis. Positive feedback also occurs when an increased hormone level further increases the synthesis and secretion of that same hormone. An example of this kind of positive feedback is found in the female reproductive system. The cyclic rise of estradiol levels provides positive feedback on the anterior pituitary and hypothalamus, causing a subsequent increase in the gonadotropin-releasing hormone and follicle-stimulating hormone. So here's that figure of feedback loops, your general model for control and negative feedback to hypothalamic-pituitary target organ systems. Your negative feedback regulation is possible at three levels: the target organ, the ultra-short feedback, your anterior pituitary, the short feedback, and the hypothalamus itself, the long feedback. So, if we're looking at the central nervous system as a positive, and it talks to the hypothalamus releasing hormone and factors release inhibiting hormones and factors to the anterior pituitary gland. The anterior pituitary gland speaks to tropic hormones. The tropic hormones go to target organs, and those target organs then release further hormone, which cause the physical effect. So we have the ultra-short feedback loop. Immediately, once the physiologic effect happens and goes back to that target organ. We also have that ultra-short feedback loop when a tropic hormone is released and lets the anterior pituitary know, hey, it's in the system, going towards the target organ. We also again have that short feedback loop at the hormone level when the target organ has released that hormone, because the anterior pituitary gland knows. It also again occurs when your tropic hormone that's supposed to go to your target organ is released, because your hypothalamus knows, hey, that tropic hormone is out. And that super long feedback happens again once the target organ releases the hormone and the hypothalamus lets it know, hey, it's out in the system. We're going to have that physiologic effect. So how do hormones transport? Once hormones are released into the circulatory system, they are distributed throughout the body. The protein peptide hormone are water-soluble and generally circulating in free, unbound forms. Water-soluble hormones generally have a short half-life of seconds to minutes because they are catabolized by circulating enzymes. For example, insulin has a half-life of 3 to 5 minutes and is catabolized by insulinases. Your lipid-soluble hormones, such as cortisol and adrenal androgens, are transported bound to a carrier protein and can remain in the blood for hours and even days. Only free hormones, those not bound to a carrier protein, can initiate changes within a target cell. At the cell membrane, your lipid-soluble hormones dissociate from the carrier protein and diffuse into the cell. Because there is an equilibrium between the concentrations of free hormones and hormones bound to plasma proteins, a significant change in the concentration of binding proteins can affect the concentration of free hormones in the plasma. So here at this table, we're looking at the binding proteins, the hormone, the factors that increase binding protein levels, and factors that decrease the binding protein. So for instance, we take the corticosteroid binding globulin. It releases two hormones, cortisol and progesterone. Cortisol gets increased via estrogen and gets decreased by the liver, or liver disease. Here's sex hormone binding globulins. That's your dihydrotestosterone, your testosterone, and your estradiol. Factors that increase that binding are none. However, factors that decrease that binding are your androgens for your dihydrotestosterone, your hypothyroidism for testosterone, and liver disease for estradiol. Your thyroid binding globulin, that's your thyroxine T4, and your triiodothyronine. Your T4 gets increased via estrogen, but gets decreased from testosterone. Your T3 gets increased by your hyperthyroidism, but gets decreased by your glucocorticoids. They also get decreased by liver disease. Your albumin, that's your all lipid-soluble hormones. They all get increased by estrogen, but can all be decreased via liver disease, malnutrition, or renal disease, so your kidney function. Your hormone receptors. Although a hormone is distributed through the body, only target cells with specific receptors for that hormone are affected. Target cell response depends on the blood level of the hormone, the concentration of target cell receptors, and affinity of the receptor for the hormone. Hormone receptors of the target cell have two main functions: to recognize and bind with high affinity to their particular hormones, and to initiate the signal to appropriate intracellular effectors. Again, we'll see Chapter 1 for cell signaling pathways. The sensitivity or affinity of the target cell to a particular hormone is related to the concentration of receptors per cell. The more receptors, the higher the affinity, or the more sensitive the cell is to the stimulating effect of the hormone. Low concentrations of hormone increase the number of receptors per cell, called upregulation. A high concentration of hormone decrease the number of affinity of receptors, called downgrading or downregulation. Thus, the cell can adjust its sensitivity to the concentration of the signaling hormone. The receptors of the plasma membrane are continuously synthesized and degraded, so that changes in receptor concentration may occur within hours. Various physiochemical conditions also can affect both the receptor number and the affinity of hormone for its receptor. Some of these physiochemical conditions are the fluidity and structure of the plasma membrane, that pH, the temperature, ion concentration, diet, and the presence of other chemicals, like your drugs. Finally, mutations in receptor structure can affect target cell activation, such as that normal cellular response are increased or decreased. For example, mutations in thyroid-stimulating hormone receptors can lead to resistance to thyroid hormone and can contribute to defective thyroid hormone production. So again, you can have the upregulation when there's a hormone and the time increases, so then your hormone receptors increase. And eventually it becomes a target cell because it has all those receptors. And then there is always that down regulation, where you see, again, through time, that hormone with all the receptors start to decrease, and the target cell has less receptors. So the regulation of target cell sensitivity, again, picture A, low hormone level and upregulated or an increase in the number of receptors, and picture B, high hormone level and downregulation or a decrease in the number of receptors. Hormone receptors may be located in or on the plasma membrane, or in the intracellular compartment of the target cell. Your water-soluble hormones are proteins that are polarized with a high molecular weight and cannot diffuse across the lipid layer of the cell or plasma membrane. They interact or bind with receptors in or on the cell membrane and activate a second messenger to mediate short-acting responses. Most lipid-soluble steroids diffuse freely across the plasma and nuclear membranes and bind to the cytosolic or nuclear receptors. Other receptors of some lipid-soluble hormones are also located in or on the plasma membrane. So again, we have, for instance, here your fat-soluble hormone that goes into the intracellular receptor, or your water-soluble hormone, those peptides. They have to go through a secondary messenger, your protein kinase A or C, to get a cellular response. So that's your hormone binding at a target cell. Your water-soluble hormone receptors. Water-soluble hormone binding with the plasma membrane receptor initiates a complex cascade of intracellular effects. In this cascade, the hormone is termed the first messenger. The hormone receptor interaction initiates a signal that generates a small molecule inside the cell, called the second messenger. The second messenger conveys the signal from the receptor to the cytoplasm and nucleus of the cell and mediates the effect of the hormone on the target cell. Your second messenger includes your cyclic adenosine monophosphate, your CAMP, cyclic guanosine monophosphate, your cGMP, and calcium, your inositol triphosphate, your IP3, and the membrane-associated diacylglycerol, your DAG, and the tyrosine kinase system. So again, here you have your second messengers identified for some specific hormones. That cyclic AMP has effects on your adrenocorticotropic hormone, your luteinizing hormone, the human chorionic gonadotropin, your follicle-stimulating hormone, your thyroid-stimulating hormone, your antidiuretic hormone, your thyrotropic-releasing hormone, the parathyroid hormone, and glucagon. Your cyclic GMP is only associated with the atrial natriuretic peptide. Calcium is associated with angiotensin II, your gonadotropin-releasing hormone, and antidiuretic hormone. IP3 and DAG also affect your angiotensin II, your antidiuretic hormone, and your luteinizing hormone, releasing hormone. The only one that's not associated with a hormone is your tyrosine phosphorylation. It's not specific to any specific or associated hormone. Your tyrosine kinase is associated with insulin, and your JAK stat is all associated with your growth hormone, your leptin, and your prolactin. So here we're taking a look at the picture. It shows examples of first and second messenger signaling. A non-steroid hormone, the first messenger, binds to a fixed receptor of the target cell. The hormone receptor complex activates that G protein. The activated G protein reacts with the guanosine triphosphate, or GTP, which in turn activates the membrane-bound enzyme adenylyl cyclase. Adenylyl cyclase catalyzes the conversion of adenosine triphosphate, your ATP, to cyclic adenosine monophosphate, your CAMP, that secondary messenger. CAMP activates protein kinase. Protein kinases activate specific intracellular enzymes, and these activated enzymes then influence specific cellular reaction and metabolic pathways, thus producing the target cell response to the hormone, ACTH, or adrenocorticotropic hormone. So just follow us down the line in order to get our end product. When the first messenger, for instance that adrenocorticotropic hormone and thyroid-stimulating hormone, from the anterior pituitary gland bind to a cell membrane receptor, intracellular levels of CAMP increase. The second messenger, that CAMP, activates protein kinases, leading to phosphorylation of cellular proteins. This either activates or deactivates intracellular enzymes, thus directing the action or products of specific cells. For example, binding of epinephrine to a beta-adrenergic receptor subtype activates the enzyme adenylyl cyclase. Adenylyl cyclase catalyzes the conversion of adenosine triphosphate, that ATP, to the second messengers, the 3 and 5 CAMP. Elevation of CAMP activates the enzyme CAMP-dependent protein kinase A, or pKa. pKa phosphorylates and activates nuclear transcription factors, the CAMP response element binding to Crib proteins, that influences numerous cellular function. Alterations in Crib activity have been implicated in many disease states, including diabetes and cancer. The actions of CAMP are terminated by the enzyme phosphodiesterase 3, which hydrolyzes CAMP into inactive adenosine monophosphates. So your cGMP also functions as a second messenger, following receptor binding of first messengers. Those hormones and signaling molecules play a crucial role in cardiovascular and pulmonary health and disease. Thus, drugs such as the phosphodiesterase inhibitors that sustain the action of cGMP are being explored for the treatment of vascular and pulmonary hypertension and cognitive dysfunction. Your hormone receptor binding of the first messenger angiotensin II and antidiuretic hormone result in generation of second messenger DAG or IP3 and triggers the release of intracellular calcium as another second messenger. Increased intracellular calcium levels can lead to the formation of calcium calmodulin complex, which mediates the effects of calcium on intracellular activities that are crucial for cell metabolism and growth. For example, calmodulin-dependent protein kinases control intracellular contractile components, your myosin and actin, which cause muscle contraction, alter plasma membrane permeability to calcium, and regulate the intracellular enzyme activity that promotes hormone secretion. Some hormone-binding messengers, such as insulin, growth hormone, and prolactin, bind to surface receptors in the cell membrane that have a transmembrane domain and directly activate second messenger of the tyrosine kinase family. These tyrosine kinases transfer a phosphate group from ATP to a protein in a cell, including the Janus family of tyrosine kinases, or JAK, and signal transducers and activators of transcription, the STAT, which makes the JAK-STAT pathway. They regulate a wide range of intracellular processes that contribute to cellular metabolism, immunity, growth, apoptosis, and oncogenesis, and are targeted for inhibition and treatment for tuning down immune-mediated responses, as in rheumatoid arthritis and cancers. So, what does that mean for us? Your steroid lipid-soluble hormone receptors, for instance. With the exception of thyroid hormones, the lipid-soluble hormones are synthesized from cholesterol, giving rise to the term steroid. These include your glucocorticoids, androgens, estrogens, progestins, mineralocorticoids, your vitamin D, and retinoid. Because they are relatively small, nonpolar, lipophilic, hydrophobic molecules, steroid hormones can cross the plasma and nuclear membrane by simple diffusion. They bind with cytosolic or nuclear receptors, which keep them from diffusing back out of the cell. Examples of cytosolic steroid hormone receptors include those for glucocorticoids and androgens, whereas progesterone has nuclear receptors. The hormone receptor complex then binds to a specific region in the deoxyribonucleic acid, or DNA, which initiates transcription of specific genes to form messenger ribonucleic acid, with translation at the ribosome for protein synthesis. Modulation of gene expression can take hours to days. So here we're taking a look at steroid hormone signaling mechanism, that lipid-soluble steroid hormone molecules detach from the carrier protein and pass through the plasma membrane. Your hormone molecules then diffuse into the nucleus, where they bind to the receptor to form a hormone receptor complex. This complex then binds to a specific site on a DNA molecule, triggering transcription of the genetic information encoded there. The resulting messenger ribonucleic acid, your RNA, molecule moves to a cytosol, where it is associated with a ribosome, initiating synthesis of a new protein. This new protein is usually an enzyme or a channel protein, produces specific effects on target cells. The classic genomic action is typically slow. Steroids also may exact rapid effects by binding to receptors on the plasma membrane and activating an intercellular second messenger. So I'll leave that up for just a moment. In addition to lipid hormone receptors for estrogen, thyroid hormone and aldosterone are also located in the plasma membrane and are associated with rapid responses from seconds to minutes. These receptors, when activated, have primarily intracellular non-genomic effects. Through crosstalk, non-genomic responses and gene transcription modulate each other, allowing cells to adapt rapidly to environmental changes. So what kind of hormone effects do we have? The binding of hormones with their receptors stimulates three general types of effects: acting on pre-existing channel-forming proteins to alter membrane channel permeability, activating pre-existing proteins through a second messenger system, and finally activating genes to cause protein synthesis. Hormones affect target cells directly or permissively. Direct effects are the obvious changes in cell function that specifically result from stimulation by a particular hormone. Permissive effects are less obvious, including obvious hormone-induced changes that facilitate the maximal response or functioning of a cell. So for example, insulin has a direct effect on skeletal muscle cells, causing increased glucose transport into the cells, and the permissive effects on mammary cells, facilitating their response to the direct effects of prolactin. Some hormones have biphasic effects that are dependent on the concentration or secretion pattern of the hormone. So for example, low or physiologic levels of antidiuretic hormone, that ADH, or arginine vasopressin, stimulate renal tubular reabsorption of sodium and water. However, at supraphysiologic levels, those that can be achieved by exogenous administration, your ADH acts as a vasoconstrictor. So structure and function of the endocrine glands. Looking at the hypothalamic-pituitary axis. The hypothalamic-pituitary axis, or HPA, forms the structural and functional basis for central integration of the neurologic and endocrine system, creating what is called the neuroendocrine system. That hypothalamic-pituitary axis produces a number of releasing inhibitory hormones and tropic hormones that affect a number of diverse body functions, including the thyroid, adrenal, and reproductive functions. So here we have the pituitary hormones and their target organs. Your hypothalamus. The hypothalamus is located at the base of the brain. It is connected to the pituitary gland by the infundibulum, the pituitary stalk, to the anterior pituitary through the hypophysical portal blood vessels, and to the posterior pituitary through the nerve tract, referred to as the hypothalamo-hypophysical tract. These connections are vital to the functioning of the hypothalamic-pituitary system. The hypothalamus contains special neurosecretory cells that are like other neurons in that they have similar electrical properties, organelles, membranes, and synapses. Your hypothalamic neurosecretory cells, however, can synthesize and secrete the hypothalamic releasing hormones that regulate the release of hormones from the anterior pituitary. In addition, these cells release or synthesize the hormones ADH and oxytocin that are stored and released from the posterior pituitary gland. So here we're looking at the location and structure of the pituitary gland. The pituitary gland is located within the sella turcica of the skull's sphenoid bone and is connected to the hypothalamus by the stalk-like infundibulum. The infundibulum passes through a gap in the portion of the dura mater that covers the pituitary. The inset shows that the pituitary is divided into an anterior portion, the adenohypophysis, and the posterior portion, the neurohypophysis. The adenohypophysis is further subdivided into the pars anterior and pars intermedia. The pars intermedia is almost absent in the adult pituitary. So I'll leave that up for just a moment. So here we're taking a look at the hypothalamus, the hypophysical portal system. And I'll leave that up for just a moment. Here we're taking a look at the nerve tracts from the hypothalamus to the posterior lobe of the pituitary gland. And again, I'll leave that up for just a moment. ADH and oxytocin travel to the posterior pituitary by way of the hypothalamo-hypophysical nerve tract. Releasing and inhibitory hormones also are synthesized in the hypothalamus and are secreted into the portal blood vessels, through which they travel to their target tissue within the anterior pituitary and control the release of tropic hormones. These releasing inhibitory hormones from the hypothalamus include a prolactin inhibiting hormone, your prolactin releasing hormone, your thyrotropin releasing hormone, gonadotropin releasing hormone, hypothalamic somatostatin, growth hormone releasing hormone, corticotropin releasing hormone, and a substance P. And those are all summarized, I guess, in this following table. So we'll take a look at that. So your hypothalamic hormones. Their target tissue, surprise, surprise, is all the anterior pituitary. So they all target that. So what does your thyrotropin releasing hormone do though? It stimulates the release of thyroid stimulating hormone, your TSH. It modulates prolactin secretion. Your gonadotropin releasing hormone stimulates the release of follicle stimulating hormone and your luteinizing hormone. Your somatostatin inhibits the release of growth hormone and TSH. Your growth hormone releasing hormone stimulates the release of growth hormone. Your corticotropin releasing hormone stimulates the release of adrenocorticotropic hormone and your beta endorphin. Substance P inhibits the synthesis and release of ACTH and stimulates secretion of your growth hormone, your follicle stimulating hormone, your LH, and prolactin, your luteinizing hormone. Your dopamine inhibits the synthesis and the secretion of prolactin. Prolactin releasing hormone does just that, stimulates the secretion of prolactin. Your prolactin inhibiting hormone, again, as the word says, inhibits the secretion of prolactin. So the anterior pituitary. The anterior pituitary, or adenohypophysis, accounts for 75% of the total weight of the pituitary gland. It's composed of three regions, the pars distalis, the pars tuberalis, and the pars intermedia. The pars distalis is the major component of the anterior pituitary and source of anterior pituitary hormones. Your pars tuberalis is a thin layer of cells on the anterior and lateral portion of the pituitary stalk, and the pars intermedia is that section that lies between the two. In the adult, the distinct intermediate lobe disappears, and the individual cells are distributed diffusely through the pars distalis and pars nervosa, the neural lobe, which are part of the posterior pituitary. The anterior pituitary is composed of two main cells, your chromophobes, which appear to be non-secretory, and your chromophilins, which are considered the secretory cells of the adenohypophysis. The chromophilins are subdivided into 7 secretory cell types, each secreting one or more specific tropic hormone. So, here we're looking at the tropic hormones of the anterior pituitary and their function. So, we're looking at your adrenocorticotropic hormone, your ACTH. Its secretory cell type is corticotropic. The target organ is the adrenal gland, the cortex. And what does it do? It increases your steroidogenesis, your cortisol and androgenic hormones, your synthesis of adrenal proteins, and contributing to the maintenance of the adrenal gland. Your melanostimulating hormone. Its secretory cell type is melanotropic. The target organ is your anterior pituitary gland. And how does it function? Well, it promotes the secretion of melanin and lipotropin by anterior pituitary and makes your skin darker. That leads us to somatotrophic hormones. Your growth hormone. Its cell type, again, is somatotrophic. And the target organ is always your muscles, your bones, and your liver. And how does growth hormone function? Well, it regulates the metabolic process related to the growth and adaptation to the physical and emotional stressors. Muscle growth, increased protein synthesis, increased liver glycogenolysis, increased fat mobilization. It also can be the target of the liver itself, which induces formation of somatomedins and insulin-like growth factors that have actions similar to insulin. Your other somatotrophic hormone is your prolactin. Its secretory cell type is lactotrophic. The target organ for prolactin is your breast, and it produces breast milk during breastfeeding for children. So, your glycoprotein hormones. That's your thyroid stimulating hormone, the TSH. The secretory cell type is thyrotropic. The target organ is your thyroid gland. And what does TSH do? It increased production and secretion of thyroid hormone, increases iodide uptake, and promotes your hypertrophy and hyperplasia of thymocytes. Then we have our luteinizing hormone. The luteinizing hormone cell type is a gonadotrophic. The target organ in women is granulosa cells, and in men, it is the Leydig cells. So what does it do? What does luteinizing hormone do? It does your ovulation, your progesterone production, or your testicular growth, testosterone production. So that's your sex hormone. Your follicle stimulating hormone, or FSH, its cell type is gonadotropic. In women, again, it is the target cells in your granulosa cells, and in men, it's the Sertoli cells. So what does that do for us in the body? Well, it's your follicle maturation, your estrogen production, and your spermatogenesis. So again, your sex hormones. Then we have our beta lipoprotein hormone, and that's a corticotropic type cell, and it targets the adipose cells. And what does it do for us? Well, it's our fat breakdown and release of fatty acids. And then we have the beta endorphins, again a corticotropic cell, which affects the adipose cells and brain opioid receptors. So how does that work? It does the analgesia that may regulate the body temperature, food, and water intake. The tropic hormones targeted by the chromophylls affect the physiologic function of specific target organs. These hormones can be grouped into three categories. Your corticotropin-related hormones, such as your adrenocorticotropic hormone ACTH, and melanocyte-stimulating hormones, your MSH. Glycoproteins, your luteinizing hormone, your follicle-stimulating hormone, and thyroid-stimulating hormone. And finally, the somatotropins, your growth hormone and prolactin. The corticotropin-related hormones are all derived from the precursor pro-opiomelanocortin, or POMC. Your melanocyte-stimulating hormone promotes the pituitary secretion of melanin, which darkens skin color. The beta-lipotropin and beta-endorphins are corticotropic hormones, also released from the anterior pituitary. Your beta-lipotropin plays a role in fat catabolism, and B and beta-endorphins impact pain perception, body temperature, and food and water intake. Your glycoproteins hormones, your follicle-stimulating hormone, and luteinizing hormone influence reproductive function and are discussed in chapter 24. Your adrenocorticotropic hormones, ACTH, regulates the release of cortisol from the adrenal cortex. Thyroid-stimulating hormone regulates the activity and thyroid gland. The roles of ACTH and TSH are discussed later on in this chapter. Again, these were all just discussed in that table. Your growth hormone and prolactin are called the somatotropic hormones and have diverse effects on the body tissues. Growth hormone is controlled by two hormones from the hypothalamus. Your growth hormone releasing hormone, which increases growth hormone secretion, and your somatostatin, which inhibit growth hormone secretion. Growth hormone is essential to normal tissue growth and maturation and peaks during adolescence. Growth hormone also impacts aging, sleep, nutritional status, stress, and reproductive hormones. In the bone, growth hormone stimulates epithelial growth and increases osteoclast and osteoblast activity, resulting in increased bone mass. Your growth hormone also increases amino acid transport in muscles. Other functions of growth hormone include your lipolysis, an enhancement of the hepatic protein synthesis. Many of the anabolic functions of growth hormone are mediated, at least in part, by the insulin-like growth factors, which are also known as your somatomedins. There are two primary forms of IGF, IGF-1 and IGF-2, so those insulin-like growth factors, of which IGF-1 is the most biologically active. They both circulate bound to a group of IGF-binding proteins, also known as IGF-BPs, modulating their activity. Your IGF-1 binds to IGF-1 receptors, mediating the anabolic effects of growth hormone. Your insulin-like growth factor 1 also binds to insulin receptors, providing an insulin-like effect on skeletal muscle. Your insulin-like growth factor 2 has an important effect on fetal growth, but suppresses growth hormone in the adult. Because of the anabolic effect of growth hormone and insulin-like growth factor 1, they can be used to treat growth disorders, increase muscle mass, and potentially slow the aging process. However, their use also has been linked to the increased rates of cancer. So, prolactin. Primary functions to induce milk production during pregnancy and lactation. It has immune stimulatory effects and modulates immune and inflammatory response with both physiologic and pathologic reactions. Its synthesis and release are increased by stimulation of the nipples and mammary gland during nursing. Your vasoactive intestinal polypeptide, serotonin, and growth factors all stimulate the synthesis of prolactin, while its release is inhibited by dopamine. Your posterior pituitary. The embryonic posterior pituitary, or neurohypophysis, is derived from the hypothalamus and is composed of three parts: the median, eminence located at the base of the hypothalamus, the pituitary stalk, and that infundibular process, also known as the pars nervosa or neural lobe. The median eminence is composed largely of nerve endings of axons that arise primarily in the ventral hypothalamus. It often is designated as part of the posterior pituitary, but contains at least 10 biologically active hypothalamic-releasing hormones, as well as the neurotransmitters dopamine, norepinephrine, serotonin, acetylcholine, and histamine. The pituitary stalk contains the axons of neurons that originate in the supraoptic and paraventricular nuclei of the hypothalamus. Axons originating in the hypothalamus terminate in the pars nervosa, which secretes the hormone of the posterior pituitary. The posterior pituitary secretes two polypeptide hormones, ADH, also known as arginine vasopressin, and oxytocin. These hormones differ by only two amino acids. They are synthesized along with their binding proteins. The neurophyins in the supraoptic and paraventricular nuclei of the hypothalamus. They are packed or packaged into secretory vesicles and are moved down the axons of the pituitary stalk to the pars nervosa for storage. The posterior pituitary thus can be seen as a site for both storing and releasing hormones, synthesized in the hypothalamus. The release of ADH and oxytocin is mediated by a cholinergic and adrenergic neurotransmitter. The major stimulus to both ADH and oxytocin release is glutamate, whereas the major inhibitory input is through gamma-aminobutyric acid, that GABA, before the release into the circulatory system. ADH and oxytocin are split from the neurophytins and are secreted in unbound form. Your antidiuretic hormone, the major homeostatic function of the posterior pituitary, is the control of plasma osmolality, as regulated by ADH. At physiological levels, ADH acts on the vasopressin 2 receptor of the renal tubule cells to increase permeability. This increased permeability leads to increased water reabsorption into the blood, thus concentrating the urine and reducing serum osmolality. These effects may be inhibited by the hypercalcemia prostaglandin E and hypokalemia. The secretion of ADH is regulated primarily by the osmoreceptors of the hypothalamus, located near or in the supraoptic nuclei. As plasma osmolality increases, these osmoreceptors are stimulated. The rate of ADH secretion increases more. Water is reabsorbed from the kidney, and the plasma is diluted to its set point. Osmolality, your ADH, has no direct effect on electrolyte levels, but with increasing water reabsorption, serum electrolyte concentration may decrease because of a dilutional effect. ADH secretion also is increased by changes in intravascular volume, which are monitored by baroreceptors in the left atrium and in the carotid arteries and aortic arch. The volume loss of 7 to 25% acts through the receptors to stimulate ADH secretion. Stress, trauma, pain, exercise, nausea, nicotine, exposure to heat, and drugs such as morphine also decrease ADH secretion. ADH secretion decreases with a decrease in plasma osmolality, an increase in intravascular volume, hypertension, an increase in estrogen, progesterone, and angiotensin II levels, and alcohol ingestion. Your physiological levels of ADH do not significantly affect vessel tone. However, at pathophysiologically high serum levels, ADH acts on vasopressin 1, receptors causing vasoconstriction resulting in an increase in arterial blood pressure. For example, high doses of ADH, given as the drug vasopressin, may be administered to achieve homeostasis during hemorrhage and to raise blood pressure in shock states. Your oxytocin. Oxytocin is responsible for contraction of the uterus and milk ejection in lactating women and may affect sperm motility in men. In both sexes, oxytocin has an antidiuretic effect similar to that of ADH. In women, oxytocin is secreted in response to suckling and the mechanical distension of the female reproductive tract. Oxytocin binds to its receptors on a myoepithelial cell in the mammary tissues and causes contraction of those cells, which increases intramammary pressure and milk expression, the letdown reflex. Oxytocin also acts on the uterus to stimulate contraction. Uterine contractions act in a positive feedback loop to further increase oxytocin secretion. Oxytocin functions near the end of the labor to enhance the effectiveness of contraction, promote delivery of the placenta, and stimulate postpartum uterine contraction, thereby preventing excessive bleeding. The function of this hormone is discussed in more detail in Chapter 24. The pineal gland. The pineal gland is located near the center of the brain and is composed of a melatonin-secreting photoreceptive cell. It is innervated by noradrenergic sympathetic nerve terminals controlled by pathways within the hypothalamus. Melatonin release is stimulated by exposure to dark and inhibited by light exposure. It is synthesized from tryptophan, which is first converted to serotonin and then to melatonin. Melatonin regulates circadian rhythm and reproductive systems, including the secretion of gonadotropin-releasing hormones and the onset of puberty. It also plays an important role in immune regulation and is postulated to affect the aging process. Further effects of melatonin include increasing nitric oxide release from blood vessels, removing toxic organ-free radicals, and decreasing insulin secretion. Melatonin has been used therapeutically in humans to help with sleep disturbances, jet lag, and inflammatory and physiologic disorders. Its utility for numerous other disorders, including anti-cancer therapy, is being explored. Your thyroid and parathyroid glands. The thyroid gland, located in the neck just below the larynx, produces hormones that control the rates of metabolic process throughout the body. The four parathyroid glands are located near the posterior side of the thyroid and function to control serum calcium levels. So here you have your epiglottis, your hyoid bone, the larynx, superior parathyroid gland, thyroid glands, inferior parathyroid gland, and your trachea. I'll leave that up for just another moment. Your thyroid gland. The two lobes of the thyroid gland lie on either side of the trachea, inferior to the thyroid cartilage, and are joined by a small band of tissues termed the isthmus, or isthmus, which crosses the anterior surface of the trachea and larynx at the cricoid cartilage. The pyramidal lobe is superior to the isthmus. The normal thyroid gland is not visible on inspection, but it may be palpated on swallowing, which causes it to be displaced upward. The thyroid gland consists of follicles that contain follicular cells surrounding a viscous substance called colloid. The follicular cells synthesize and secrete thyroid hormone. Neurons of the autonomic nervous system terminated on the blood vessels within the thyroid gland and on the follicular cells themselves. Acetylcholine, catecholamines, and other peptides directly affect secretory activity on the follicular cells and thyroid blood flow. Approximately a 2-month supply of thyroid hormone is stored in the gland itself. So here we have the thyroid follicle cells, and I'll leave that picture up for just a moment. Also found in the tissue of the thyroid are parafollicular cells, or C cells. C cells secrete various regulatory peptides, including calcitonin, and, in much smaller quantities, the neuropeptides ghrelin, serotonin, and somatostatin. At high levels, calcitonin is also called thyrocalcitonin. Lower serum calcium levels by inhibiting bone resorbing osteoclasts. However, in humans, the metabolic consequence of calcitonin deficiency or excess do not appear to be significant. Your calcitonin is used therapeutically to treat a number of bone disorders, including osteoporosis, osteoarthritis, Paget bone disease, hypercalcemia, osteogenesis imperfecta, and metastatic cancer of the bone. The precursor molecule to calcitonin is called procalcitonin and is a stress hormone that is elevated in infectious and inflammatory disorders, and its measurement can aid in the diagnosis of serious diseases. The regulation of your thyroid hormone secretion. Thyroid hormone, TH, is regulated through a negative feedback loop involving the hypothalamus, the anterior pituitary, and the thyroid gland. This loop is initiated by TRH, which is synthesized and stored within the hypothalamus. TRH is released into the hypothalamic-pituitary portal system and circulates to the anterior pituitary, where it stimulates the release of TSH. TRH levels increase with exposure to cold or stress, and from decreased levels of T4. So here we have that feedback system right here. Thyroid stimulating hormone, TSH, is a glycoprotein hormone synthesized and stored within the anterior pituitary. It circulates to bind with TSH receptor sites located on the plasma membrane of the thyroid follicular cells. The primary effects of TSH on the thyroid gland include an immediate increase in the release of stored thyroid hormone, an increase in iodide uptake and oxidation, an increase in thyroid hormone synthesis, and an increase in the synthesis and secretion of prostaglandins by the thyroid. TSH also increases growth of the thyroid gland by stimulating thymocyte hyperplasia and hypertrophy and decreasing apoptosis. As TH levels rise, there is a negative feedback effect on the HPA to inhibit TRH and TSH release, which then result in a decreased TH synthesis and secretion. TH synthesis also is controlled by serum iodide levels and by circulating selenium-dependent enzymes called your iodinases, which inactivate the precursor molecule thyroxine. Your thyroid gland hormone and the regulation of function are summarized here in this table. So, our thyroxine, the T4, and triiodithyronine, the T3. Those are the two hormones. Their regulation are controlled by TSH, released in the response to metabolic demand. Influence on amount is secreted by sex, pregnancy, gonadal, and adrenocortical increased steroids, and equal the increased level of exposure to extreme cold and increased levels. Nutritional state, chemical, your growth hormone, your GHIH levels equal the decreased levels of dopamine, which equals the decreased levels of catecholamines, which those levels increase. Your functions. It regulates protein, fat, carb catabolism in the cells. It regulates your metabolic rate in all the cells. It regulates your body heat, insulin antagonist. It maintains your growth hormone secretion and skeletal maturation. It has an effect on your CNS development. It is necessary for muscle tone and vigor. Maintains cardiac rate, force and output, and maintains secretion of your GI tract. It affects your respiratory rate and oxygen utilization. It also maintains calcium mobilization, has an effect on your red blood cell production, and stimulates your lipid turnover, your free fatty acid release, and cholesterol synthesis. Your calcitonin. Instead of trying to figure out how to properly read its regulation, because it feels off somehow, the way it's presented in this little graph, so I'll just read you the function of calcitonin instead. Calcitonin lowers serum calcium by opposing bone resorption effects of PTH, your prostaglandins and calciferol by inhibiting osteoclastic activity. It lowers your serum phosphate levels, may also decrease calcium and phosphorus absorption in the GI tract. So, the synthesis of your thyroid hormone. Thyroid hormone synthesis is summarized in the following steps. An uniodinated thyroglobulin is produced by the endoplasmic reticulum of the follicular cells. That's step 1. Step 2. Your tyrosine is incorporated into the thyroglobulin as it is synthesized. Third step, your iodide is actively transferred from the blood into the colloid by carrier proteins located in the outer membrane of the follicular cells. This active transport system is called the iodide trap and is very efficient at accumulating the trace amounts of iodide from the blood. Step 4. The iodide is oxidized and quickly attaches to tyrosine within the thyroglobulin molecule. Step 5. Coupling and iodinated tyrosine forms thyroid hormone. Your triiodothyronine, your T3, is formed from the coupling of the monoiodide tyrosine and diiodotyrosine. A tetraiodotyrosine, or T4, is commonly known as thyroxine, and that's formed by coupling of two diodotyrosines. So pretty much the coupling happens at that point for either T3 or T4. Last step. Your thyroid hormones are stored attached to thyroglobulin within the colloid until they are released back into the circulation. So the thyroid gland normally produces 90% of T4 and about 10% of T3. Once released into the circulation, both T3 and T4 are primarily transported bound to one of three carrier proteins: your thyroxine binding globulin, your albumin, or your lipoproteins. The bound form serves as a reservoir, whereas the unbound form is active. In the body tissues, most of the T4 is converted to T3, which acts on the target cells. T3 binds with different receptors: your TR alpha 1, your TR beta 1, and your TR beta 2. So, actions of thyroid hormone. TH has, or thyroid hormone has a significant effect on growth, maturation, and function of cells and tissues throughout the body. TH is essential for normal growth and neurologic development in the fetus and infant, and affects metabolic, neurologic, cardiovascular, and respiratory function across the lifespan. In addition, TH is required for the metabolism and function of blood cells, as well as normal muscles function and the integrity of skin, nails, and hair. Similar to some steroid hormones, thyroid hormone binds to intracellular receptors complexes and then influence the genetic expression of specific proteins. They also may bind to receptors on the plasma membrane, your mitochondria and cytoplasm, and mediate non-genomic effects. Thyroid hormone affects cell metabolism by altering protein, fat, and glucose metabolism, and as a result, heat production and oxygen consumption are increased. Your TH has permissive effects throughout the body, optimizing the action of other hormones and neurotransmitters. These effects can become very pronounced when there are either high or low levels of circulating thyroid hormone. For example, in the heart, your T3 stimulates the synthesis of specific contractile proteins. Your sarcolemma ion pumps, your sodium potassium ATPase pump, your calcium channel ATPase chain and pump, and membrane receptors, your beta adrenergic receptors. Therefore, in hyperthyroidism, which is associated with the elevated levels of thyroid hormone, cardiac effects include an increased heart rate and cardiac output, as well as the development of cardiomyopathy, because that heart has to just keep pumping harder and harder, causing those cells to grow. Your thyroid hormone also has effects on the respiratory center, contributing to the normal hypoxic and hypercapnic drives. In severe hypothyroidism, ventilation can become very depressed. The thyroid hormone also stimulates bone resorption, and hyperthyroidism is associated with osteopenia, hypercalcemia, and hypercalciuria. That leads us to our parathyroid glands. Normally, two pairs of parathyroid glands are present behind the upper and lower poles of the thyroid gland, but the number may range from 2 to 6. The parathyroid gland produces parathyroid hormone, your PTH, which is the single most important factor in the regulation of serum calcium concentration. The overall effects of PTH secretion is to increase serum calcium concentration and decrease the concentration of serum phosphate. A decrease in serum ionized calcium levels stimulates PTH secretion. On release, PTH enters the circulation in unbound form. The hormone attaches to plasma membrane receptors in target tissues, where the biologic effects of PTH are mediated primarily by activation of the adenylyl cyclase system. To achieve regulation of serum calcium concentration, PTH acts directly on bone with at least two effects. In acute hypocalcemia, PTH secretion stimulates osteoblasts to release receptor activator for nuclear factor, receptor activator of nuclear factor K beta ligand, and macrophage colony stimulating factor, your MCSF, which results in osteoclast proliferation, maturation, and release of acidic enzymes, such as cathepsin. These enzymes mobilize calcium release from the bone, which increase the serum calcium level. There is a bone remodeling with chronic stimulation by PTH, a process in which bone is broken down and reformed. Paradoxically, when PTH is administered intermittently at a low dose, it stimulates bone formation. This observation led to the use of PTH for treatment for osteoporosis. So here we have the normal metabolism regulated by PTH and vitamin, a macrophage colony stimulating factor, and your parathyroid hormone. So I'll leave that up for just a moment before I keep reading. PTH also acts on the kidney to increase calcium reabsorption while phosphate reabsorption is decreased. PTH acts on the plasma membrane receptor on the distal tubules of the nephron to increase reabsorption of calcium. It acts on the proximal tubules to decrease reabsorption of phosphorus and bicarbonate. The resultant increase in serum calcium concentration inhibits PTH secretion. In renal cells, PTH also stimulates the activity of 1-alpha hydroxylase, which mediates a step in the formation of the biologically active form of vitamin D, or your 1,2,5-dihydroxy vitamin D. Vitamin D works as a cofactor with PTH to promote calcium and phosphate absorption in the gut and enhances bone mineralization. Vitamin D also plays an important role in metabolic processes and controlling inflammation, and has been found to be deficient in the majority of individuals in the United States and many other countries. Magnesium and phosphate concentrations also affect PTH secretion. Hypomagnesemia in persons with normal calcium levels act as a mild stimulant to PTH secretion, whereas in hypocalcemia individuals, hypomagnesemia decreases PTH secretion. Your hyperphosphatemia leads to hypocalcemia because of calcium phosphate precipitation in soft tissue and bone. Alterations in serum phosphate level therefore may indirectly influence PTH secretion by affecting serum calcium. Another hormone that plays an important role in calcium and bone physiology is the parathyroid hormone releasing protein, This hormone is synthesized in many adults and fetal tissues and affects tissues around it in a paracrine fashion. It has a similar biologic property to PTH and uses the same receptors but has other actions mediated by different regions within the molecule. It is important for endochronal bone formation and bone remodeling. It was discovered as a primary hormone involved in a malignancy-related hypercalcemia. Your endocrine pancreas. The pancreas is both an endocrine gland that produces hormones and an exocrine gland that produces digestive enzymes. The pancreas is located behind the stomach between the spleen and the duodenum and houses the islets of Langerhans. The islets of Langerhans have four types of hormone-secreting cells: alpha cells, which secrete glucagon; beta cells, which secrete insulin and amylin; delta cells, which secrete gastrin and somatostatin; and F or PP cells, which secrete pancreatic polypeptide that stimulates gastric secretion and antagonizes cholecystokinin. These hormones regulate carbohydrate, fat, and protein metabolism. Nerves from both of the sympathetic and parasympathetic divisions of the autonomic nervous system innervate the pancreatic islets. So here we're taking a look at the pancreas. And I'll leave that up for just a moment, and then I'll start reading again. The perfusion of the anterior lobe of the pancreas, where alpha, beta, delta, and most numerous comes from the branches of the superior mesenteric artery. The posterior lobe is perfused by branches of the celiac artery. The pancreatic islets receive 10% of the pancreatic blood flow, but represents 1% of the pancreatic mass. It is necessary for oxygenation and the delivery of the islet hormones to target cells. That leads us to insulin. The beta cells of the pancreas synthesize insulin from the precursor proinsulin, which is formed from a larger and earlier precursor molecule, preproinsulin. Proinsulin is composed of an A peptide and a B peptide connected by a C peptide and two disulfide bonds. C-peptide is cleaved by proteolytic enzymes, leaving the bonded A and B peptide chain that become insulin. Insulin circulates freely in the plasma and is not bound to a carrier. C-peptide can be measured in blood as an indirect measure of serum insulin synthesis. The secretion of insulin is regulated by chemical, hormonal, and neural control. Insulin secretion is pulsatile, increasing when the beta cells are stimulated by the parasympathetic nervous system, usually before eating a meal. Other factors stimulating insulin secretion include increased blood glucose of glucose amino acids and gastrointestinal hormones. Insulin secretion diminishes in response to low blood levels of glucose, high levels of insulin, and sympathetic stimulation of beta cells in the islets. Prostaglandins also inhibit insulin secretion. At the target cells, insulin binds with an enzyme-linked plasma membrane receptor that contains tyrosine kinase on the cytosolic surface found on cells throughout the body. Insulin receptors binding sends a cascade of signals to activate glucose transporters for entry of glucose into the cells. The primary glut is called GLUT4. It is stored in cellular vesicles until activated by insulin receptors and is then translocated to the cell surface, where it facilitates the diffusion of glucose into the cell. The translocation of GLUT4 to the cell surface is associated with a 10 to 21-fold increase in glucose diffusion into the cell, particularly skeletal and cardiac muscle, liver, and adipose cells. Insulin sensitivity, denoting the biologic responsiveness to insulin, is affected by age, weight, abdominal fat, and physical activity. Your insulin resistance, a subnormal biologic response to normal insulin concentrations, has been implicated in numerous diseases, including hypertension, heart disease, and type 2 diabetes. Your adipocytes release a number of hormones that are altered in obesity and have an important adverse effect on insulin sensitivity. The most effective measures shown to improve insulin sensitivity in humans is weight loss and exercise. So here we're taking a look of how insulin action on the cells begins. Insulin is an anabolic hormone that promotes glucose uptake, primarily in the liver, muscle, and adipose tissue. It also increases the synthesis of proteins, carbohydrates, lipids, and nucleic acids. The following table summarizes the actions of insulin. The net effect of insulin in these tissues is to stimulate protein and fat synthesis and decrease blood glucose levels. The brain, red blood cells, kidneys, and the lens of the eye do not require insulin for glucose transport. Insulin also facilitates the intracellular transport of potassium, phosphate, and magnesium. Insulin is metabolized in the liver and kidney by enzymes that split disulfide bonds. Very little insulin is excreted, unchanged in urine. So again, here we see how insulin actions work, where they work, in the liver cells and muscle cells or adipose cells, and whether there is an increase or a decrease. So I'll leave that up. Amylin. Amylin, islet amyloid polypeptide, is a peptide hormone co-secreted with insulin by beta cells in response to nutrient stimuli. It regulates blood glucose concentration by delaying gastric emptying and suppressing glucagon secretion after meals. Amylin also has a satiety effect, which reduces food intake. Through these mechanisms, amylin has an anti-hyperglycemic effect. Your glucagon. Glucagon is produced by the alpha cells of the pancreas and by cells lining the gastrointestinal tract. Glucagon is an antagonist to insulin and acts to increase blood glucose during fasting, exercise, and hypoglycemia. It acts primarily in the liver to increase blood glucose concentration by stimulating glycogenolysis. Glucagon also stimulates gluconeogenesis in the kidneys and lipolysis in adipose tissue. Muscle glucagon is used only by muscle and not systematically. High glucose levels cause glucagon release to be inhibited. Low glucose levels and sympathetic stimulation promote glucagon release. Amino acids such as alanine, glycine, and asparagine also stimulate glucagon secretion. A protein-rich meal has the same effect. Glucagon also stimulates lipolysis, which has a ketogenic effect, caused by the metabolism of free fatty acid in the liver. These effects have led to the hypothesis that impaired glucagon secretion is as important as insulin insufficiency in the pathogenesis of diabetes. Your pancreatic somatostatin. Pancreatic somatostatin is produced by delta cells of the pancreas in response to food intake and is a hormone essential in carbohydrate, fat, and protein metabolism. It differs from hypothalamic somatostatin, which inhibits the release of growth hormone and TSH. Pancreatic somatostatin is involved in regulating alpha cell and beta cell function within the islets by inhibiting secretion of insulin, glucagon, and pancreatic polypeptide. Your incretins, the incretin hormone, are secreted from endocrine cells in the gastrointestinal tract in the presence of your carbohydrates, proteins, and fats. The major incretin hormone are glucagon-like peptide 1, your GLP-1, and glucose-dependent insulinotropic polypeptide, your GIP. They control postprandial glucose levels by promoting glucose-dependent insulin secretion, inhibiting glucagon synthesis, promoting hepatic glucose secretion, and delaying gastric emptying. Your incretins also enhance beta cell mass and replenish intracellular stores of insulin. Incretins are broken down by an enzyme called dipeptidyl peptidase 4, and drugs that inhibit the enzyme increase incretin levels. The gliptins are incretin agonists used for the treatment of type 2 diabetes. So that leads us to gastrin, ghrelin, and pancreatic polypeptides. Your gastrin is released by G cells in the stomach and stimulate the secretion of gastric acid. Ghrelin is an intestinal hormone and stimulates growth hormone secretion, controls appetite, and plays a role in obesity and the regulation of insulin sensitivity and glucose tolerance. Your pancreatic polypeptide is released by PP cells, your gamma cells, in response to hypoglycemia and protein-rich meals. It promotes gastric secretion, antagonizes cholecystokinin, and is frequently increased in the pancreatic tumors and diabetes. Your adrenal glands. The adrenal glands are paired pyramid-shaped organs located behind the peritoneum and close to the upper roll of each kidney. Each gland is surrounded by a capsule embedded in fat and well supplied with blood from the phrenic and renal arteries and the aorta. Venous return from the left adrenal gland is to the renal vein and from the right is to the inferior vena cava. Each adrenal gland consists of two separate portions, an outer cortex and an inner medulla. These two portions have different embryonic origins, different structures, and different hormonal functions. The adrenal cortex and medulla function like two separate but interrelated glands. So, here is the structure of the adrenal gland, showing cell layers of the cortex. And I'll leave that up for just a moment. The adrenal cortex. The adrenal cortex accounts for 80% of weight of the adult gland. The cortex is histologically subdivided into the following three zones. The zona glomerulosa, the outer layer, contributes about 15% of the cortex and primarily produces the mineralocorticoid aldosterone. The zona fasciculata, or the middle layer, constitutes about 78% of the cortex and secretes the glucocorticoids cortisol, cortisone, and corticosterone. And then finally, the zona reticularis, that inner layer that constitutes about 7% of the cortex and secretes the mineralocorticoids, your aldosterone, adrenal androgens and estrogen, and glucocorticoids. The cells of the adrenal cortex are stimulated by the anterior pituitary hormone, ACTH. All hormones of the adrenal cortex are synthesized from low-density lipoprotein cholesterol. The best-known pathway of steroidogenesis involves the conversion of cholesterol to pregnenolone, which is then converted to the major corticosteroids. Your glucocorticoids. The glucocorticoids are steroid hormones and have metabolic, neurologic, anti-inflammatory, immunosuppressive, and growth-suppressing effects. They are released under stress conditions. They act through nuclear, genomic, and non-genomic membrane-initiated steroid signaling pathways in the cells. The term glucocorticoid refers to those steroid hormones that have direct effect on carbohydrate metabolism. These hormones increase blood glucose concentration by promoting gluconeogenesis in the liver and conserving glucose by decreasing uptake of glucose into the muscle cells, adipose cells, lymphatic cells, by antagonizing insulin. This results in increased glucose for the brain during stress. In extrahepatic tissue, the, the glucocorticoids stimulate protein catabolism and inhibit amino acid uptake and protein synthesis. The ultimate effect on the body is protein catabolism and muscle wasting. So here we're taking a look of the effects of the glucocorticoids on the body. I'll leave that up for just a moment longer and we'll keep right on going. The glucocorticoids act at several sites to suppress immune and inflammatory reactions. They affect innate immunity through several pathways, including the inhibition of antigen presentation by dendritic cells and decreased activity of pattern recognition receptors on the surface of macrophages. They decrease the proliferation of T lymphocytes, primarily the T helper lymphocytes, and there is a greater adverse effect on T helper 1 cell cytokine production than there is on the T helper 2 cell cytokine production, and therefore greater depression of cellular immunity than humoral immunity. Your glucocorticoids also decrease immune and inflammatory response by decreasing the natural killer cell activity, by blocking phospholipase A and the synthesis of prostaglandin, thromboxanes, and leukotrienes, and by inhibiting inflammatory gene expression. In addition, glucocorticoids suppress synthesis, secretion, and action of chemical mediators involved in inflammatory and immune responses, including histamine, adhesion molecules, inducible cyclooxygenase, and inducible nitric oxide synthase. The glucocorticoids increase resistance to the severe inflammatory response to lipopolysaccharides through the inhibition of cytokines, chemokines, certain hormones, and neurotransmitters. In addition, glucocorticoids stimulate anti-inflammatory cytokines. Lysosomal membranes are also stabilized, decreasing the release of proteolytic enzymes. The suppression of innate and adaptive immunity by glucocorticoids means that infection and poor wound healing are some of the most problematic complications of the use of glucocorticoids in the treatment of disease. Similarly, psychologic and physiologic stress increases glucocorticoid production, which provides a pathway for the well-described decrease in immunity seen in both acute and chronic stress conditions. Other effects of glucocorticoids include the inhibition of bone formation, inhibition of ADH secretion, the stimulation of gastric acid secretion. Your glucocorticoids appear to potentiate the effects of catecholamines, including sensitizing the arterioles to the vasoconstrictive effects of norepinephrine. Thyroid hormone and growth hormone effects on adipose tissue also are potentiated by glucocorticoids. The metabolite of cortisol may act like a barbiturate and depress nerve cell function in the brain, accounting for the noted effects on mood associated with steroid level fluctuation in disease or stress. Your pathologically high levels of glucocorticoids increase the number of circulating erythrocytes, increase their appetite, promote fat deposition in the face and cervical areas, increase uric acid excretion, decrease serum calcium levels, suppress the secretion and synthesis of ACTH, and interfere with the action of growth hormone so that somatic growth is inhibited. The most potent of naturally occurring glucocorticoids is cortisol. It is the main secretory product of the adrenal cortex and is needed to maintain life and regulate the body during both positive and negative stress. Cortisol has biologic half-life of approximately 90 minutes, with the liver primarily responsible for its deactivation. Cortisol secretion is regulated primarily by the hypothalamus and the anterior pituitary gland. Your corticotropin-releasing hormone is produced in several nuclei in the hypothalamus and stored in the median eminence. Once released, your CRH travels through the portal vessel to stimulate the production of ACTH, beta-lipotropin, gamma-lipotropin, endorphins, and enkephalins by the anterior pituitary. ACTH is the main regulator of cortisol secretion and adrenocortical growth. So here you are looking at the feedback of the glucocorticoid synthesis. The precursor from which ACTH is synthesized is pro-opiomelanocortin, or POMC. Three factors appear to be the primarily involved in the regulation and regulating the secretion of ACTH. The negative feedback effects of high circulating levels of cortisol and synthetic glucocorticoids suppress both CRH and ACTH, whereas low cortisol levels stimulate their secretion. Your diurnal rhythms affect ACTH and cortisol level. Whether in person with regular sleep-wake patterns, ACTH peaks 3 to 5 hours after sleep begins and declines throughout the day. And cortisol levels follow a similar pattern, peaking just before awakening. And your psychologic and physiologic hypoxia, hypoglycemia, hyperthermia, and exercise. Stress increases ACTH secretion, leading to an increased cortisol level. There also is evidence that there is synthesis and secretion of glucocorticoids from extra-adrenal tissue, including the thymus, lung, intestine, skin, brain, and possibly the heart in response to immune stimulation. This is thought to provide autocrine and paracrine immune regulation and control of inflammation. When ACTH is secreted, it binds to specific plasma membrane receptors on the cells of the adrenal cortex and on other extra-adrenal tissues. Because both adrenal and extra-adrenal tissues have ACTH receptors, a number of effects result from stimulating or the stimulation by ACTH. So here you'll see the effects of the adrenocorticotropic hormone on adrenal effects and the extra-adrenal effects. Once ACTH stimulates the cell of the adrenal cortex, cortisol synthesizes and secretion immediately occurs. In the healthy person, the secretory pattern of ACTH and cortisol are nearly identical. After secretion, most cortisol circulates in bound form, but 15 to 30% is bound to albumin, 55 to 75 is tightly but reversibly bound to a plasma glycoprotein called transcortin, and about 10 to 15% is unbound. The levels of transcortin play a role in the HPA feedback system, controlling cortisol secretion. Transcortin levels are significantly elevated by increased estrogen level that occur with pregnancy and hormone therapy. The unbound portion is free to diffuse into cells, but only those with specific intracellular glucocorticoids receptors respond to cortisol stimulation. Your ACTH is rapidly inactivated by the circulation, and the liver and kidney remove the deactivated hormone. Your your mineralocorticoids, such as your aldosterone. Your mineralocorticoid steroids directly affect ion transport by epithelial cells, causing sodium retention and potassium and hydrogen loss. Aldosterone is the most potent of the naturally occurring mineralocorticoids and acts to conserve sodium by increasing the activity of the sodium pump of the epithelial cells in the nephron. The initial stages of aldosterone synthesis occurs in the adrenal zona, fasciculata, and zona reticularis. The final conversion of corticosterone to aldosterone occurs in the zona glomerulosa. Aldosterone synthesis and secretion are regulated primarily by the renin-angiotensin-aldosterone system, although other factors also may be involved. The renin-angiotensin system is activated by sodium and water depletion, increased potassium levels, and a diminished effective blood volume. Angiotensin II is the primary stimulant of aldosterone synthesis and secretion. However, increases in serum potassium concentration also directly stimulate aldosterone secretion. ACTH acutely stimulates aldosterone secretion, but is secondary to angiotensin II and potassium. So here is the feedback mechanism regulating aldosterone secretion. When sodium and potassium levels are within normal limits, approximately 50 to 250 milligrams of aldosterone is secreted daily. Of the secreted aldosterone, 50 to 75% bind to plasma proteins. The proportion of unbound aldosterone contributes to its rapid metabolic turnover in the liver. Its low plasma concentration and its short half-life, about 15 minutes, aldosterone is degraded in the liver and is excreted by the kidneys. Aldosterone maintains extracellular volume and blood pressure by acting on distal nephron epithelial cells to increase sodium reabsorption and potassium and hydrogen excretion. The renal effect takes about 90 minutes to 6 hours. Other effects of aldosterone include the enhancement of cardiac muscle contraction, stimulation of ectopic ventricular activity through secondary cardiac pacemakers in the ventricles, stiffening of blood vessels with increased vascular resistance and decreased fibrinolysis. Your pathologically elevated levels of aldosterone have been implicated in the myocardial changes associated with heart failure. Your adrenal estrogens and androgens. The healthy adrenal cortex secretes small amounts of estrogen and androgens. ACTH appears to be the major regulator. Some of the weak androgenic substances secreted by the cortex are converted by peripheral tissues to stronger androgens, such as testosterone, thus accounting for some adrenergic effects initiated by the adrenal cortex. Peripheral conversion of adrenal androgens to estrogen is enhanced in aging or obese persons, as well as in those with liver disease or hyperthyroidism. The biologic effects of metabolism of the adrenal sex steroids do not vary from those produced by the gonads. Your adrenal medulla. The adrenal medulla, together with the sympathetic division of the autonomic nervous system, is embryonically derived from neural crest cells. Chromaffin cells are the cells of the adrenal gland that store and secrete the catecholamines epinephrine and norepinephrine. Both are synthesized from the amino acid phenylalanine. Only 30% of circulating epinephrine comes from the adrenal medulla. The other 70% is released from nerve terminals. The medulla is the only minor source of norepinephrine. The adrenal medulla functions as a sympathetic ganglion without postganglionic processes. Sympathetic cholinergic preganglion fibers terminate on the chromaffin cells and secrete catecholamines directly into the bloodstream. The catecholamines are therefore hormones and not neurotransmitters. So here you see the synthesis of catecholamines. Physiologic stress to the body, your traumatic injury, hypoxia, hyperglycemia, and many other triggers the release of adrenal catecholamines through acetylcholine, which depolarize the chromaffin cells. Depolarization causes exocytosis of storage granules from the chromaffin cells, with release of epinephrine and norepinephrine into the bloodstream. Secretion of adrenal catecholamines is also increased by ACTH and the glucocorticoids. Once released, the catecholamines remain in the plasma for only seconds to minutes. The catecholamines exert rapid biologic effects after binding to plasma membrane receptors, your alpha 1, alpha 2, beta 1, beta 2, beta 3 in target cells, and activating the adenylyl cyclase system. Catecholamines are rapidly removed from the plasma by neurons for storage in new cytoplasmic granules, or they may be metabolically inactivated and excreted in urine. Catecholamines have diverse effects on the entire body. Their release and the body's response have been characterized as the fight or flight response. In general, the effects of catecholamines activate adrenergic receptors on cell membranes of all visceral organs and smooth muscle and promote hyperglycemia. So, tests of endocrine function. The evaluation of the endocrine system is challenging because 1. The complexity of the clinical presentation as a result of multiple organ system involvement, and 2. The nonspecific nature of complaints frequently associated with endocrine dysfunction, and 3. The inappropriate use of laboratory test interpretations. Tests of the endocrine system involve several general types of clinical evaluations. Measurement of hormone level is accomplished by radioimmunoassay, by enzyme-linked immunoobservant assay, and less commonly by bioassay. Your radioimmunoassay (RIA) is a technique for measuring the minute quantities of hormone in the blood. Uses antibodies and radiolabeled hormones to determine the quantity of hormone in the plasma. Your enzyme-linked immunosorbent assay, your ELISA, also is used to determine circulating hormone levels. This method is similar to that of RIA, but is less expensive and easier to conduct. Instead of a radio-labeled hormones, an enzyme-labeled hormone is used. A bioassay involves the use of graded doses of hormone in a reference preparation and then comparison of the results with an unknown sample. Bioassays are used more commonly in investigative endocrinology than in clinical laboratories. If the serum level is greater or less than the reference values, more definitive tests are required to determine the source of the problem. Measurements of individual hormones does not always permit differentiation between normal and abnormal values when hormone levels are changing over time. For an accurate interpretation, the broad normal range of some hormones requires knowledge of previous hormonal levels and timed sampling. Stimulation and suppression tests that determine the response to exogenous stimulants or inhibitory can help to decipher some of these complexities. Indirect assessment of hormonal function often includes measurements of concentrations of serum glucose and electrolytes that are affected by the endocrine process. Evaluation of hormonal function also may include radiographic imaging of specific glands. So, that leads us to aging and the endocrine system. The precise relationship between aging and the endocrine system is not clear. Perhaps most important, the question of whether changes in endocrine function are a consequence or a cause of aging has yet to be resolved. These relationships have been difficult to identify, in part because the number of age-related variables that may coexist, such as acute or chronic non-endocrine disease, use of medications, alterations in diet, body composition and weight, and changes in sleep-wake cycles. However, the endocrine system is so integral to health that changes in endocrine function have been used as biomarkers for unhealthy aging. Investigation into the role of the endocrine gland and their interactions in the aging process has generated much data, although the evidence is contradictory. There are complex changes within the HPA, altered biologic activity of hormones, altered circulating levels of hormones, altered secretory response of the endocrine glands, altered metabolism of hormones, loss of circadian control of hormone secretion, and effects of oxidative stress and inflammation are among the findings. Changes in secretion of hypothalamic regulatory factors and hormones, or changes in the hypothalamic feedback sensitivity may contribute to alterations in control of an optimal internal environment. The dynamic equilibrium of the endocrine system also may be affected by altered secretion of neurotransmitters within certain areas of the brain, affecting hypothalamic and pituitary function. Such as alterations may include an excess or deficit in secretion of pituitary hormones and loss of appropriate secretory pattern of those hormones. Loss of endocrine steady states may be associated with or contributing to aging. The most studied change in hormone function with aging affect the levels of the reproductive hormones and gonadotropins, such as your menopause and andropause. Two of the most important endocrine changes associated with aging affect the thyroid gland and the pancreas. Other important hormone changes associated with aging include the decline in serum levels of growth hormone and IGF-1, parathyroid hormone, the dehydroepiandrosterone, and ADH. Your thyroid gland. Changes in thyroid structure and function occur with aging. Structurally, some glandular atrophy and fibrosis occur, with nodularity and increasing inflammatory infiltrates. These infiltrative changes may reflect age-related autoimmune damage. Clinical signs of thyroid disease are more difficult to detect in older adults. Overall, it is estimated that there is some evidence of thyroid dysfunction in 5 to 10% of older adult women. The presence of thyroid nodules increases after the age of 70. Changes relative to thyroid hormone and its function are more difficult to assess, and much of the available data is contradictory. Overall, TSH secretion is thought to increase slightly, but there is controversy regarding whether this is normal aging or the presence of hypothyroidism, with risk for adverse consequences. Your treatment for thyroid deficiency is also affected by aging. The appropriate dose for TH, or thyroid hormone, replacement is often lower in adults because the peripheral metabolism of TH decreases with age. In addition, TH must be replaced slowly in older adults with coronary artery disease to prevent angina and myocardial infarcts. The endocrine pancreas. It is estimated that 40 to 50% of individuals older than the age of 65 have impaired glucose tolerance or diabetes, and there is an age-dependent decline in beta cell function. With aging, pancreatic cell regeneration declines, and there is an increased distribution of fat tissue. Dysfunction of the pancreas with decreased insulin secretion of beta cells and insulin receptors and increased insulin resistance have all been documented and may be related to changes in adipokine physiology. These changes have significant implications for many target organs, particularly the cardiovascular system, which is increasingly at risk for both vascular and cardiac disorders. Your growth hormone and insulin-like growth hormone factors. The amounts of growth hormone and insulin-like growth hormone decline with aging, a process that has been called the somatopause. This decline in anabolic stimuli is linked to a decrease in muscle size and function, decreased amounts of fat and bone mass, and changes in reproductive and cognitive function. Some studies have shown that growth hormone resistance and declines in growth hormone are associated with longevity in animals, but this has not been confirmed in humans. Your parathyroid glands. The age-related alteration in PTH secretion has been proposed to explain alteration in calcium homeostasis that have been noted in older adults. Calcium intake, especially in women, tends to decrease with aging and may contribute to osteoporosis. The average daily intake of 450 to 500 milligrams per day causes a negative calcium balance greater than 40 milligrams per day and may be related to the absolute bone loss of approximately 1.5% per year. Older adults show decreased intestinal adaptation to variations in calcium intake. Hyperparathyroidism may occur secondary to calcium malabsorption and hypocalcemia, with increased bone remodeling that result in cortical bone thinning and porosity. Elevated levels of parathyroid hormone have been linked with an increase in mortality in older adults, many of whom have a persistent hypercalciuria, which indicates a defective renal mechanism for responding to decreased calcium intake. Decreased circulating levels of vitamin D are common in older adults, especially those in long-term care institutions. Vitamin D deficiency has been linked to not only osteoporosis but also cancer, autoimmune disease, diabetes, cardiovascular disease, and mental disorders. The parathyroid gland, kidney, and choroid plexus secrete the clotho-protein, which, when overexpressed, has anti-aging effects. The adrenal glands. The adrenal cortex loses some weight and has more fibrous tissue after the age of 50. Age does not appear to affect the feedback mechanism involved in maintaining glucocorticoid levels, but there is an age-related decrease in the metabolic clearance rate of the glucocorticoids. The metabolic clearance of cortisol decreases with an age-related decline in liver and kidney function. Further, less cortisol appears to be used by the body when aging is accompanied by a loss of lean body mass. Decreased clearance and reduced use of cortisol contribute to higher circulating cortisol levels, but diurnal variation is maintained. Because feedback mechanisms are intact, the higher cortisol levels cause a decrease in cortisol secretion. Circadian pattern of ACTH and cortisol secretion may change with aging. Your plasma levels of the adrenal androgens, as well as the urinary excretion of the metabolic end products, decrease gradually but dramatically with age, to as much as 50 to 70% of young adults. This change in adrenal function has been called the adrenopause and is correlated with a decreased synthesis activity of DHEA. This change appears to reflect a decline in the function of the zona reticularis in postmenopausal women. This decline in adrenal androgen secretion is especially important because nearly all sex steroids after menopause come from adrenal and ovarian production of androgen precursor converted to estrogen in the periphery. In older adult men, adrenal androgen production accounts for more than half of the circulating testosterone levels. And lastly, the antidiuretic hormone. Although hyponatremia is a common finding in older adults, it appears related to changes in renal function or sensitivity rather than the ADH-related mechanisms. Morphologic studies have not shown significant age-related degenerative changes in the neuroendocrine pathway that regulates the synthesis and secretion of ADH. However, it appears that ADH secretion is augmented when stimulated by changes in osmotic concentration, whereas baroreceptor-mediated ADH secretion is reduced, producing the syndrome of inappropriate ADH secretion, common in older adults. And there it concludes our chapter. We will continue on with chapter 22 in just a moment. So stick around. Bye.